Okay, welcome back. This is part 4 for our second lecture. The title is Fermentation and Storage of Beer. Okay, fermentation. The cold wort is inoculate with yeast in starter tank, then main tank, or we will proceed or brew in the measured amount to the variated wort. 1 pound of liquid yeast per barrel of wort. That's the quantity that use yeast inside the fermenter. Okay, yeast is in the presence of air. We metabolize sugar to carbon dioxide and water. But in the absence of air, yeast will metabolize sugar to carbon dioxide and alcohol or ethanol. Okay, yeast are even used to produce beer, such as Saccharomyces cerevisiae and Saccharomyces caspergensis. Okay, for fermentation process for beer production, the operation operate near to strive condition. It's about 3 to 14 degrees Celsius, depend on yeast and brewery condition. Okay, for Saccharomyces cerevisiae, we use a top fermenter. Then the yeast itself will rise to the top upon completion, and the range of temperature is about 12 to 14 degrees Celsius, and ale is produced in a robust and complex manner. For Saccharomyces cerevisiae, we use a bottom fermenter. Flocculated and settled on the bottom upon the completion. The temperature is about 3 to 5 degrees Celsius, and then lager and beer produced by this yeast is produced smooth and clean and malty taste. Okay, this is a diagram of fermentation tank or fermentation vessels. Okay, this is a schematic presentation of the experiment gas lift bioreactor, or this is or air lift bioreactor. You can see inside the bioreactor itself we have an internal tube. This is a gas distributor. There's no turbulence, no propeller to move the liquid. Here just using the gas. You can see they have a inoculate and a gel bit inside. That's cool. They also have a bubble inside. Okay, another one is a principal method of cell immobilization. Immobilization have four steps. The first entrainment within the matrix, and then attachment or absorption to the preformed carrier. The third one is self-aggregation of cells, and the fourth one is cell contained behind a barrier. Okay, for the fermentation, specific gravity fall when yeast grow, ferment the growth of wort. Okay, and then the fermentation complete about 9 days. In wort, 3 to 7% alcohol content per volume. The fermentation lower the pH to 4 and produce 0.3% of dissolved carbon dioxide. Okay, this is condition for if the person drink a lot of beer. This is called beer belly, and it's very harmful to the body and very harmful to the liver. Okay, after fermentation, the beer quickly chill to 0 degrees Celsius, pass through a filter, and to remove excess yeast and other suspended material that pump into pressure storage tank for several weeks or months. It will allow green or young beer to age and settling for final suspended protein, yeast cell, and other remaining material. Development in this process also will happen the development of ester and other flavor and contribute to improve body and more mellow flavor. Okay, this is a storage tank used to storage or to age the beer. Okay, for finishing steps, after storage, it is given last fine filtration to yield sparkling clear beverage. Further carbonated to 0.5% carbon dioxide, and fill into cast, bottle, or cans under pressure of carbon dioxide to prevent gushing, foaming, and keep air out. If this step is if the step is if the beverage is not sterile, hops and mild acidity provide mild preservative, so it cannot be contaminated. And pasteurization also at 60 degrees Celsius for several minutes also is not sterile, but it will prolong the shelf life of the beverages. Okay, this is a finishing step that we just discussed. It's a filtration unit, flash pasteurization, and packaging using a can, or this one is a cat.